the kidneys chapter. Okay. All right. Okay. Within the kidneys, we talked about their function, right? What is their function to do? What is their function? What's the function of the kidneys?
Filtration.
Filtration, right? And are they filtering blood or are they filtering the plasma in the blood?
Blood?
No, plasma.
Plasma, because yes, what is blood? What is it?
Red blood cells are not getting filtered out. I mean, they're getting filtered out, but they don't go through the actual filtration process, right? They just get separated. They go right back into circulation. It's the plasma that gets filtered out, right? The plasma gets separated from the red blood cells and goes into the tubules to remove any waste and contaminants, right? And then within the structures of the kidneys, we know that we have two kidneys, and there's different regions within the kidneys. There's the cortex, which is that outer portion of the kidney, and then the medulla, right? And if you teased out the actual medulla, you can see that these little small blood or small vessels, small tubes, are organized in a way where they're organized in a way where they create the actual diamond-shaped medulla. Okay, so you can see the medulla contains most of the loops of Henle and these distal and proximal tubules. Okay, if we zoomed in further and looked at the functional unit of the actual kidney, right, the actual part of the kidney that is doing the actual filtration, that's called the nephron. Okay, and the nephron looks like this little kind of like convoluted tubule structure. Okay, and is the kidney, is the nephron itself, is that kind of a standalone tubule structure, or is it connected to blood vessels and arteries and veins?
Connected.
It's highly connected, right? So we're delivering blood to those nephrons, and then we're also recollecting the blood that has been filtered out, right? And we're either picking up or dispensing more substances like water or salts and ions, right? So here, this is how it's kind of organized, right? So one part of the nephron is organized in the cortex, the other one is in that medulla region, right? So, and again, these tubules, right, including the proximal and distal convoluted tubules, they are surrounded in close proximity to these capillaries that are called the protrubular capillaries. Okay, and so those capillaries are collecting and recollecting and dumping off salts, collecting more water, depending on what the body needs, right? And aside from just filtration, what does the kidney do to your body's homeostasis? What are two things it balances out in your body?
pH.
pH, yes. What else?
Water?
Water content, yes. Good job. Yes. It changes your pH and water content, right? If you're dehydrated, you'll recollect water from those the filtration from the nephrons, right? If you're overly hydrated, right, you'll urinate more, right? What is the process of urination called? What's the technical word for it?
Micturition.
Micturition, yeah.
That's right.
And when you have an imbalance in your pH, right, the kidneys try to balance that pH off by reabsorbing certain chemicals, dispensing buffers, right, into the bloodstream so that you balance out your pH.
Wait.
So, can you guys turn off your thing? What is it called?
Oh, sorry.
Yeah. All right. And so the actual anatomy of the kidneys, right? Within the nephron, we have the Bowman's capsule, right? It's that end part of the tube, right? And that Bowman's capsule is surrounding a little portion of those blood vessels, right? And those blood vessels reside within the Bowman's capsule, and that is called the glomerulus, okay? And so as blood comes in, it diffuses out of this glomerulus into the Bowman's capsule, and you filter out that plasma. Once the plasma is filtered, it's called something else. Do you guys remember what that's called?
No, I don't recall.
Filtrate?
Yeah, filtrate. Yeah, so it becomes filtrate, and then you go into the proximal descending loop of Henle, ascending loop of Henle, and then distal tubule. Within each of these different sections of tubes, you can do one of a couple of different things, right? Filtration happens mostly in this region of the Bowman's capsule, right? And then there are two other functions that we're doing. What are the two other functions that we can do aside from filtration? Or three, actually.
Secrete hormones.
What are the
secretion?
Secretion, right? So we can secrete things into the blood to modulate homeostasis. We can reabsorb, right? So we can reabsorb some nutrients, right? Reabsorb water, and then excretion, right? So removing the substances out of the out from within the body, right? So within the proximal convoluted tubule, right, we're doing reabsorption and secretion. In the loop of Henle, we only do one process, and what process is that? Reabsorption, right? And then back into the distal tubule, we are doing both reabsorption and secretion, right? Cool. And so this diagram kind of breaks down the different portions of those, the anatomy of the nephron and their function. And what exactly is doing the filtration? How does the filtration happen? Like, what is the science behind it? Yes, so. Diffusion, right? And you're passing all that plasma through a screen, right? Like a screen made up of cells. So we have these podocytes that are wrapping around the glomerulus, those arteries, that afferent arteries are coming into that Bowman's capsule. And they're screening and filtering the blood, right? They're filtering the plasma and they're moving it into this capsule, okay? That then flows into the proximal tubule. Now, does the filtration rate matter?
Yes.
Yeah, absolutely, right? If you, for a normal individual, you guys remember that?
Damn it.
Like total per day?
Yeah.
180 mils?
Liters.
Oh,
Not
liters.
Liters, yeah. So 180 liters, right? So that's like what, 62-liter bottles, right? And so what could happen if you decrease the filtration rate? What accumulates? What happens in your body? What happens to the organs?
They don't work
They start shutting down?
Yeah, they don't get enough blood.
Toxins build up?
More importantly, the toxins build up, right? You start accumulating waste, right? You accumulate waste, you change the pH of your body, right? And you're not able to maintain blood pressure. Do you guys remember that exercise, the scenario-based exercise? Right? We talked about what happens to blood pressure, right? During renal failure, right? And also potentially sugar, right? Sugar or blood glucose levels. So there's a lot of things that can happen, right? If your kidneys don't maintain that regular filtration rate. And what is that filtration rate called? What is the name for it?
Is it the GFR?
Yeah, glomerular filtration rate. Yes. Perfect. All right, and this is just a diagram showing you the actual slits that the cells are making, right? And how they're organized around those capillaries, right? So here you can see the capillary walls, the lumen, right? And the podocytes are kind of wrapping around that capillary, right? And how are these cells right here? They're shown here, right? There's these bundle of cells that are associated with the proximal tubule, the loop of Henle here. And then they're also associated to the efferent and afferent arterioles. What do these cells do?
I don't remember.
These here. You guys remember the function of these cells? Remember, if you have blood coming into the afferent arteriole, dumping it, all that blood and plasma into the Bowman's capsule, and then the blood flowing out, right? What happens is the flow of blood, right, slows down here. What do these cells do?
Damn.
How do they regulate the blood flow?
They open.
Yeah, they can either speed it up or slow it down to maintain that constant GFR, right? So blood flow is decreased in through the afferent arteriole. We can vasoconstrict these blood vessels, increasing the flow, right? If we have too much flow, if it's too fast, we can vasoconstrict these blood vessels, right? So these are specialized cells that act to modulate and control blood flow. Let's see what they're called. They're called mesangial cells. And the process is called tubular glomerular feedback, right? You have these cells that can recognize the flow of blood, right? So here you have granular cells secrete this chemical called renin. It's a hormone that can modulate water balance in your body, right? And you have these cells called the macula densa cells right against that loop of Henle. And so they're detecting the blood flow, right? They're detecting the GFR, and depending if the GFR increases or decreases, is going to constrict or vasodilate these blood vessels. And it does it by sending out chemical signals called paracrine signals, okay? And this is just showing you that juncture glomerular apparatus where, you know, if GFR increases, right, that means you're having too much blood coming in, right, too fast. The flow through the tubular structure here, the loop of Henle, increases, right? You're having too much blood flow, or plasma in that case, right? And so these cells, the macula densa cells, can detect that blood flow, right, or filtrate flow, right? Just be accurate. And they will release the chemical signals that can then constrict the afferent arteriole. Okay. All right. So the processes that the kidneys can go through is reabsorption, right? So reabsorption can be active and passive, right? If it's active, you need ATP. If it's passive, you're just using the normal diffusion osmosis, right? No energy needed. And so active transport, right, usually uses sodium ions, right, to create a difference in the concentration of sodium. And so if you're trying to bring water into the cell, you're going to use energy to pump sodium into the cell so that the water molecule follows, right? So this is a couple of diagrams showing you how we can reabsorb things, right? So let's say this is the extracellular fluid. This is the tubule lumen of the kidney, right? Of the nephron. And let's say we want to reabsorb water into the extracellular fluid, meaning back into the bloodstream. Well, in order to do that, we're going to increase salt concentration in the extracellular fluid so that the water molecules follow, right? Does that make sense for you, to y'all?
So basically, the salt, wherever salt is going, the water will flow behind
Water will
it.
flow.
Okay,
yeah.
I see.
So you're reabsorbing the water in that sense,
I
right?
see.
And so sometimes you need to use energy to do that. Other times you don't, right? And in this case, the kidneys, salt matters. Salt is that, like, you can imagine salt in this case is like the stock exchange. It's the bonds, right? They're going to be the things that dictate where the water goes, right? So reabsorption, let's see, we talked about secretion, right? So secretion, that is the active movement of molecules from the extracellular fluid into the nephron. Okay, so now we're taking things from the outside, from the blood flow, back into the nephron, right? So it's important for homeostatic regulation of hydrogen ions and potassium. And so what do these things do? How does hydrogen ion affect your body? If you increase hydrogen ions in your body, what happens to your pH?
Lowers.
It increases. But it does, yes, you're right. It affects it. It actually increases it. I'm sorry, lowers. Sorry. Did you say lower?
Yeah.
I'm sorry. Yes, sorry, lowers it. Yeah. So it becomes more acidic. Sorry, that's my bad. Right. So your body becomes acidic, the pH lowers, right? It decreases, it goes to 0. If your body's pH is more basic, it's higher, right? It goes towards 14, right? So yes, so the increasing secretion increases nephron excretion, right? So it can also influence how much, urine you're removing, right? And so again, there we showed some examples of how organic substances can affect the secretion of things, right? So specifically molecules that can change and modulate the pH. Lastly, excretion, right? So how much are you peeing, right? The excretion part is what you pee, right? What you're urinating. And the total amount of excretion is equal to how much you're filtering out and how much you're reabsorbing. So you kind of subtract that from the total equation. And then plus how much you have secreted into the lumen, right? So clearance is a non-invasive way of measuring GFR, right? Sometimes GFR, you have to, use certain catheters and measure cell function, but just by looking at how much you microturate, I guess, or P, you can look at GFR function, right? And lastly, here's a snapshot showing how your kidneys handle certain substances and what they do and where are they reabsorbed or secreted, right? So in the proximal tubule, you're mostly reabsorbing salts, ions, and glucose, right? And then in the ascending loop, again, you're reabsorbing these salts, but then you're secreting out urea, right? And urea, again, is like a metabolic waste, right? And the distal tubule, again, you're removing salts, ions, calcium. And lastly, in the collecting duct, the duct that's right before going into your bladder, you're reabsorbing any more salts and ions, urea, and then you could also absorb certain substances. I think that's all we went through. We we did talk about microtuition, the actual, the spinal reflex that control microtuition, right? So, and we have two different types of neurological control, right? We have simple spinal reflexes, right? And so that just means that you're gonna have these stretch receptors and the autonomic control of your bladder, right? But the reflexes can be overridden or taken over by your normal processing, right? So remember that the example I said, we're not horses where we just kind of like pee everywhere, right? Even if you have the urge to pee, you have some control of overriding that urge to microtuate and hold your urine, right? And that is due to the presence of both simple spinal reflexes, right? And much more complicated reflexes under motor control, okay? Any questions before we go on?
I got a question,
Yeah.
Professor.
Mm-hmm.
So for this quiz that's coming up, is there any labeling that we need to understand? Like, labeling that's going to be on a quiz?
Labeling? No, a lot of it's just like function. Like if it's labeling, it'll probably be like which one of these portions along the tubules undergoes filtration, right? And either you'll have to label it or match it and stuff like that. But I think for the most part, it's just going to be just straight multiple choice questions and stuff like that. But you should definitely know this. Like, this is the important part, right? Because you could have questions saying, okay, where is most of the filtration happening, right? You have to say Bowman's caps or which processes like reabsorption, where does reabsorption mostly happen, right? And you would be in the loop of Henle or the end of the loop of Henle, right? So stuff like that. Anything else?
No.
All right.
And you said on the quiz, in the first sort, it's the kidneys, the
digestive, immune, and then like a portion of the reproductive cycle.
What about the rest of respiratory? Was that on there?
Which one?
The respiratory, something like that.
I thought we already got tested on that.
We did respiratory. It was immune system, digestive, reproductive, and there was one more.
Immune.
Yes, there you go.
Yeah. We'll go through all those.
Oh, that's what I meant, reproductive. Oh, my bad.
Yeah, reproductive system, yeah. So now the digestive system, right? So we talked about the digestive system. I hope the digestive system was pretty easy. It wasn't too difficult, right? Like I don't know. It seems like it's one of the easiest ones, right? The digestive system runs from one end all the way to the other. So it's basically one large tube right through your center mass, right? And it has both like mechanical and chemical processes that are happening, right? And along the entire tube, what is called your gastrointestinal system, you have these little stops or things called sphincters, where they stop the bolus of food, right? And it's like a gateway that it has to go through, right? Before it goes to the next process. So if we start from our mouth, we're breaking down food mechanically. We're using some enzymes to chemically digest the food and break it down, right? And then it goes into the stomach. And in the stomach is going to have that first barrier, right? You're going to have a sphincter there that basically stops the food from going straight into your intestines. And in your stomach, what is the main process of the stomach? Like, what are we doing in the stomach?
Getting all the nutrients and stuff, breaking down the food.
Breaking down the food, yes, you're breaking down food in the stomach, right? It's basically a container for your food to be mixed in with hydrochloric acid, right? It's just a holding tank, right? Like imagine like you have a car, right? the gas tank just holds the gas, right? Doesn't do anything with the gas. It's until the gas goes into the injection system where you actually use the gas, right? So let me see, where's the stomach? Okay, so let's just go before we get into stomach. The digestive function, right? There's 4 main functions. You're going to digest the food, right? Break it down. You're going to absorb the food. You're going to undergo some secretions to secrete out chemicals, enzymes that it can help you metabolize that food. And then you also have to move that food down the digestive tract, right? So some challenges for your stomach or your intestine, your digestive system to overcome is auto digestion, right? When you eat food, you have to find a way to avoid eating yourself in the process of eating and digesting food, right? Sometimes when we're eating, like we don't realize you sometimes bite yourself, right? And then even within your stomach, right? If you have like these ulcers in your stomach, they can break the lining of your stomach up where you will start digesting your own stomach, right? So we have to avoid autodigestion. We have to maintain balance. So if we're eating 10 pounds of food, right, we have to account for that 10 pounds. How much of that food are we using to build ourselves up? And then how much of that food is being removed as waste, right? It has to equal the mass balance law. And then defense, right? So we're eating so many different types of things. Microorganisms make their way into our digestive tract. We need to have a way of protecting ourselves from those things. And so here's just kind of like a snapshot of like those 4 processes. So you have food that's being broken down in digestion. At the same time, that food is undergoing motility. You're moving it down your GI tract. Your cells within that tract are going to secrete chemicals that will then break the food down further. And some cells in your digestive tract will absorb that food, right? So along your GI tract, you have smooth muscles, right? And so that smooth muscle is used to undergo contraction so that you can move the food down your digestive tract. And the 2 types of muscle contraction that we have are called tonic and phasic contractions, right? So tonic contractions happen within minutes and hours, whereas phasic contractions happens in seconds, right? So these 2 contractions work together to both mix and move the food around in your stomach so they can kind of mix up and come into contact with the enzymes that you're secreting out, and also move the food down the digestive tract. So here you're showing you just kind of like the migrating pattern of the food, right? The food drops down into the stomach, right? Goes into the small intestines and then is moved throughout the small intestine where you absorb the nutrients. Hopefully by the time it gets into the small intestines, the food has been broken down to its individual nutrients, right? Its amino acids, its monomer sugars, right? All those like biomolecules we talked about. And so here's a diagram showing you those different types of motility movements, right? So when you have the bolus of food moving down your GI tract, it could go through peristaltic contractions, right? Where you can just squeeze one end and push the food in one direction, or through these kind of phasic contractions, you could kind of mix the food around back and forth, sloshing it around, mixing it, right? And those are called segmental contractions. They alternate back and forth so you can move the food around. All right, and so your GI tract is actually controlled by your nervous system. It has some type of association with your nervous system, right? And so that is called the enteric nervous system. And it can act independently. So it has some control of motility, secretion events, and then also growth of the digestive system, right? So your digestive system is dynamic, and so it could grow, it could get stronger, it could get weaker, right? What are some ways that your digestive system can get weaker or not function as properly? Like, what are some foods that you can eat that can kind of change your digestive system?
Fiber.
Fiber, yeah. So you have a low fiber diet, right? And you only eat soft, liquid food. What do you think is going to happen to your digestive system? How do you think it's going to adapt to that?
It's not getting
It's gonna get soft too.
nutrients.
Yeah, it's going to get soft too, right? You're
Yes.
going to need those phasic contractions, right? It's probably not going to dominate during digestion, right? And in your stomach, right? Maybe you don't create the same amount of acids and enzymes, right? What about if you're eating a really tough, hard food, like high fiber, like your body's gonna adapt to, right?
Constipation.
Yeah. Well, for fiber, hopefully you're drinking water, right? Yeah, but if you're drinking hard fibrous food, yeah, you can get constipated if you're not drinking enough water, right? What about people that go back and forth between having an all plant diet and an all meat diet?
They tear stuff up.
They tear it up, right? You don't support, if you're eating all plant fibers and all plant diet, you're not supporting bacteria that live in your gut, right? That will break down that meat. You'll be supporting the life of bacteria that can break down like plant material and stuff like that, right? And so your microbiome, like the life that is living within you, right? The bacteria changes. And so if you switch over right back to eating meat, you're going to get those bubble guts. Remember that?
Yeah.
You don't have the enzymes, and the bacteria are not accustomed to digesting that meat. And so you're going to be like, oh, it's going to take you a while to get accustomed to that, right? Yeah. Okay, so we talked a little bit about these hormones that are very common and control different processes of your digestive system, right? So in your stomach, you release this hormone or this chemical called gastrin, right? And the primary effect is that it stimulates acid production, right? So you're going to have receptors in your stomach that will detect that food and release gastrin, right? In your intestines, you're going to have these other chemicals: CCK, secretin, motilin, GIP, and GLP-1. And each of those has a function in stimulating digestion, stimulating absorption, or suppressing appetite and satiety, right? So GLP-1 is a big thing right now. It's a hot drug, right? And it helps people with diabetes lose weight and control their appetite, control their blood sugar. Because GLP-1, glucagon-like protein, it's a protein that's like glucagon, which is a hormone, right? And what does glucagon do as a hormone?
Provide energy.
Hold the sugar.
Yeah. And so this GLP-1 peptide can stimulate insulin release, right? It inhibits glucagon release, and then it affects the gastric function, right? It affects your appetite and your secretions of all these digestive enzymes. So make sure you know the basic functions of these, right? Because they'll show up, and we'll talk about what is the function of GLP-1. So it promotes satiety, it stimulates insulin release, right? What is the function of motilin? It stimulates migrating motor complexes and it controls the movement of food, right? Let's see what we talked about. Swallowing, we talked a little bit about swallowing, right? And swallowing, you have during the process of swallowing and mastication, right? That is the first step in digestion, right? You're mechanically breaking down the food with your teeth, right? And what else is happening there? What are you secreting out in your mouth, in your saliva?
Enzymes?
What enzyme did we talk about?
Amylase.
Amylase. Did you say amylase?
Yeah.
Yeah, amylase, right? Amylase is an enzyme that can start breaking down complex sugars into smaller sugars, right? And so if you're eating sweets or eating bread, right, you can start breaking down those complex sugars into smaller, easier to digest and absorb sugars. Yeah. Let's see, what else did we talk about?
I got a question.
Yeah, go ahead.
So we said amylase. Does that also contain where, like, you know how people accidentally swallow gum?
Yeah.
That only breaks down like the flavor and the sugar of it, and it doesn't break down like the whole gum.
Well, yeah, but like gum is different though, because gum is made up of other like substances that are not sugar based. They're kind of just like made up of these polysaccharides and fibers that are not digestible. Okay, and so you can't break it down because you don't have the enzymes to break it down. So that's why they say gum is kind of tricky, because if you keep swallowing gum, it's going to get trapped in your stomach because we can't break it down, right? Gum is mixed in with other sugars for flavoring, right? Other substances to give it that flavor of mintiness and stuff like that, and oils. But the actual substance of gum is just things that we can't digest. That's why they say not to swallow it, right? But yeah. Anything else? That's all I'm gonna focus on for this. I'm not gonna go into detail of like acid secretion and stuff like that. Just the main functions of digestion, right? The mouth, the stomach, the intestines. Oh, we didn't go through this one thing, but where's most of the water being absorbed in? Is it your small intestine, your large intestines?
Large.
Large. Right. We're absorbing most of the water in the large intestine. Yeah. And then make sure you go through these important hormones, right, of the GI tract. All right.
So basically just the main functions of the digestive, respiratory, and then just hormones?
Yeah.
Okay, Professor.
Yeah. And then lastly, or not lastly, but going into this is the immune system, right? What do we talk about the immune system about? I'm into pretty much a good amount of detail, right? I want you to focus on knowing the difference between the innate and the adaptive, right? The innate immune system is fast-acting, non-specific. There's no memory. It's made up of physical barriers, chemical barriers, and mechanical barriers, right? It's all right here. And it's basically encompasses some cells. I want you to know the general function of these cells, okay? What do macrophages do?
They swallow up the viruses?
Yeah, viruses or bacteria, right?
Yes.
And then natural killer cells, what is their function?
They can kill themselves.
They kill your own cells that are either virally infected or cancerous, right? And then basophils and eosinophils are cells that are kind of sentry cells that can release histamine during allergic reactions or release cytokines, right, to induce an immune response. And then the adaptive immune system, right, it's slow, it's specific to one type of pathogen, and there's a memory component. And you have to adapt, right? It requires your adaptive immune system requires for you to come into contact with that organism that's, going to get you sick in order to develop some type of immunity to it. And within the adaptive immune system, there's two halves. There's the cell-mediated responses and the antibody-mediated responses, right? Or also, we could also call them the cell-mediated and the humoral immunity. In the cell-mediated, we have T cells functioning mostly. And the humoral immunity, or what they label here, the antibody-mediated immunity, we're using antibodies, right? Which of these two cells creates antibodies, T cells or B cells?
B
cells.
B cells.
Yes, B cells, yeah. And what are the different roles of T lymphocytes? What do cytotoxic T cells do?
Never mind.
They kill. They toxic, right? They'll initiate apoptosis of cells that are not normal.
That's right.
Right? What about helper T cells? What do?
They help
Alert the cavalry.
Yeah, they're the
Signal.
cavalry, right? They release the cytokines and they control other cells and activate them. Yeah. And then regulatory cells, what do they do? What do regulatory T cells do?
Regulating.
They regulate, and so they'll stop the immune system. They'll suppress the immune system and tell everybody, hey, calm down, the fight's over, right? And so if you don't have a functioning regulatory T cell population, you can develop some autoimmune disorders, right? All right. We talked about the lymphoid tissues, right? What are the two primary lymphoid tissues? The thymus and the bone marrow, right? The secondary lymphoid tissue are encapsulated lymph nodes and the spleen, right? So the lymph nodes that are surrounded are actually distributed along your body, right? And they're the sites where they sequester large amounts of lymphocytes, and they bring in all that nasty, harmful bacteria that may be outside or floating around in your body to that area, so they could fight it, right? And so it has its own lymphatic vessel system, right? So they could travel down the lymph vessels, right, and bring all that bacteria and lymph fluid back to the lymph node. And again, the lymph node is connected to your circulatory system. So whenever you need more support and more nutrition, right, blood is delivered to that lymph node. And that is why you get these engorged and swollen lymph nodes during an infection, right? Leukocytes are the immune cells, right? So white blood cells that we generally call white blood cells. They include granulocytes, phagocytes, and antigen-presenting cells, right? And those specific cell names are basophils, eosinophils, and neutrophils. Okay, so I need you to know these names and their function, okay? We also have monocytes and macrophages, right? Dendritic cells. And so far, all of these cells that I named out function in your lymphocytes, right? Lymphocytes. These are going to be the T and B cells. These cells come from a different line of cells, and they only act in the adaptive immune system, okay? Now they're developed in your primary lymphoid tissue. So T cells, where are they born?
Thymus.
they're mature in the thymus,
Oh, the
but they're born
bone
in the
marrow.
bone marrow. Yeah. So they're born in the bone marrow, just like B cells, but they mature in the thymus. Does that make sense? And then the B lymphocytes, like the B cells, they mature and they're born in the bone marrow, and they also mature in the bone marrow. Okay. And this is just showing you the lineage, right? So we have these progenitor cells giving rise to all the other white blood cells. And then you have the lymphocyte stem cell that only gives rise to the T and B cells, right? And also the natural killer cells. And we talked about the function of the immune system, right? In order to have these cells looking for things that are harmful to your body, that could give you an infection, they have to distinguish themselves from everything outside, right? So that process is called they're the self, right? Self versus non-self. So the cells are distinguishing who are foreign invaders and who are part of our body, right? And so there's a diagram here showing you that, right? So let me see, where's that diagram at? Where is it here? Here we go. So no, it's not that. What did I talk about that? Did I talk about that? Oh yeah, here we go. So this is a little late in the chapter, but I just want to talk about it, right? So we talked about how your cells are communicating with one another, how they detect pathogens and antigens, right? And so this is a diagram showing you how T cells are activated by the pieces of the antigen that the macrophage has found or swallowed up, right? So here you have this large antigen presenting cell. It just swallowed up, let's say, a bacteria, and it mounted pieces of that bacteria on its surface on these molecules called MHC class complex MHC molecules, okay? And so what they're showing you here is the T cell, it's kind of like a cop, right? Coming to this antigen presenting cell and looking at the evidence. It first looks at whether or not that antigen presenting cell is part of the body. So it's looking for the self. And then it's going to look for what it's presenting to it, right? So it's looking at the ID and the evidence, right? And so all of your cells in your own bodies have their own type of MHC class 1 molecules, right? And only T cells and antigen presenting cells contain MHC class 2 molecules because they can communicate so they with one another so that they can look out for the bad pathogen, right? Give me 1 second, I gotta turn off my alarm. All right, I'm back, sorry. Okay, so where did I leave off at? Okay, how cells communicate, right?
Yes.
Let's see. Let's take a step back. All right. All right. So cell communication, right? So once a T cell binds with an antigen presenting cell, it's going to communicate with it by using those MHC class 2 molecules, MHC class 1 molecule. And if something's wrong and it detects that an antigen on that surface of the APC, antigen presenting cell, is going to release cytokines, right? And remember, cytokines are chemical messengers that can induce an immune response. These are a couple different types of molecules that are part of the innate immune system, okay? So that non-specific process. Chemotaxins, what do chemotaxins do?
They chemo.
You guys remember that? They allow the cells to move towards the chemicals, right? So chemical taxins. Opsonins, they mark certain antigens or bacteria for destruction. Pyrogens, they induce fevers, right? And histamines induce an allergic response, okay? Now, B cells, right? Oh, here's the part about MHC class 2 molecules, right? So, major histocompatibility complexes, right? All your cells in your body, except for red blood cells, contain an MHC class 1 molecule. So it's like an ID, right? It's how they are able to distinguish and determine who's who, right? When a T cell comes knocking. An MHC class 2 molecule are found on antigen presenting cells and macrophages, right? So those are molecules that can mount different pieces of antigens and abnormal proteins so that this T cell can come by and check for, diseases and stuff like that. Now, B cells, right? B lymphocytes, they produce antibodies, right? And antibodies are proteins that these cells can secrete out and they produce. There's how many classes? 1, 2, 3, 4, 5 different classes of antibodies. The typical structure of an antibody is like this Y structure. And in this Y structure, right, you have a constant domain, this little handle here, that is always the same. And then you have 2 little Y-shaped prongs that are the variable domain. Those little Y structures here change depending on what the antigen is, okay? So they can modify their light chains and heavy chains right here. Their light chains, sorry, so that they can accommodate for many different types of antigens to detect, okay? And phagocytes, right? Phagocytes are a type of cell that can swallow things up, right? So some examples of phagocytes are macrophages, dendritic cells, neutrophils, right? They can actually swallow things up like bacteria and then utilize those pieces of the bacteria that they just broken up, right? That they tore up and mount the pieces of the bacteria on the surface of their cells. So they become antigen presenting cells, right? They're presenting the antigens that were once bound on the surface of the bacteria, on the surface of their cells. And so they're basically a walking like billboard, right? Like you ever seen those billboards at the police station where they have like wanted or whatever, like, or looking for this person or missing person, right? They're basically like that, right? The MAC class 2 molecules are those posts, okay? They're those framed for that wanted sign. And when a T cell comes by and looks at that sign, right, it's going to recognize what it's going to look for, right? And then it's going to go do the job, right? And do some type of response. So we're back to this figure. So this antigen presenting cell is presenting the perpetrator. The T cell is looking for that antigen and making sure it's a match. And then it's going to do some type of signaling. So it's going to activate itself. It's going to send out cytokines to activate other cells. And then it's going to mount an immune response. Okay. I think this is where we're going to stop because otherwise it's going to be too too detailed. Any questions?
No.
Okay. All right, let's see. Last but not least, one more. Keep it going. Come on. Hard through, people.
We got it.
All right.
You got it, Doctor. You got it, Dr. Santana.
Not me. It's you guys. I'm worried about you guys.
No, we're paying attention. We're paying attention.
Glassy-eyed. Okay. Reproduction. Okay, finally. We're at reproduction, right? Reproduction is the process of passing on your genes to your progeny, right? As humans, we have reproductive organs, sex organs, right? Within those organs, we produce gametes or seeds, right? That have half of the information to make a human being, right? In males, they contain testes that produce sperm. And in females, their gonads are ovaries that produce ova, right? And so as a human race, we're typically dimorphic, meaning we have two different sexes, right? And within those two different sexes, in those two different sexes, they have 46 chromosomes, like 46 books to make a human, right? Each sex donates half of those books, right? And it could be a different flavor of book, right? So there's going to be a pair of books, right? There's going to be 22 pairs of books. And then one of the books in that set is going to be the instructions to make a male or a female, to differentiate between a man and a female. And so the eggs and the sperms are haploid, meaning they have half the chromosomes. 22 of those chromosomes are called autosomes because they have the instructions to make everything else except sexual characteristics. And then one of those chromosomes is going to be the sex chromosome, okay? So typically, we have a chromosomal arrangement, right, that determines genetic sex as either X for female or XY for male. The Y chromosome is essential to make a male, to make male reproductive organs, okay? Now, there's some variance to that. So sometimes things get crossed over incorrectly, or you gain an extra chromosome, and that can give you different variations of sex determinants and sexual characteristics, right? So you can have XXY or XYY or just an X, right? And some of those variants and different patterns of chromosomal arrangements, some of those are not viable. They won't produce a live human, right? The fetus will die. There's going to be some differences in the patterns of development that don't produce a viable offspring, right? But some of them do. And so you're going to have these different sexual variants like Turner syndrome, right? So this is just a diagram showing you the different arrangements of those dimorphic sexual arrangements, right? So XX, you produce a female. XY, you produce a male, right? And so as viable human beings that can reproduce, you're going to create gametes, right, of these different flavors. So a female can only produce gametes with an X chromosome. Males can produce gametes with a Y chromosome and an X, right? And it's just the luck of the draw, right? You're just gambling to see which one you get, right? So the male, right, whichever sperm fertilizes the egg, the male has not the final say, but the male is the one in charge, the one that you blame, right? So they're donating that either X or Y. And here's a diagram showing you a microscope image of what the chromosomes look like. So here's 1 through 22 through 22 autosomes, right? So chromosomes that just have the instructions to make a human, right? And then one set of sex chromosomes, X and Y, right? So you can envision in each of these little ova or sperms, they're going to have half of these chromosomes, right? Which is this 22 plus one of these two. In the case of males, you're going to have an X and a Y. In the case of the female, you're only going to have XX. Okay? Does it make sense? So if I were to take an ova, an egg cell from a patient, and I were to look at it under a microscope, take out the nucleus, right, and stain these chromosomes with a special dye, this is what it looks like. If I could tease it out, this is what they'll look like, right? All right. So each individual that is developing as a fetus, right, as an embryo, starts off with bipotential gonads, meaning they have sexual organs that will become sexual organs that are developing, right? And they can develop into two ways. And the two ways to develop depends on the genes available and the hormones that are being produced, okay? So the bipotential gonads in a fetus before it reaches sexual determination include the internal structures, which are the Wolffian ducts or Mullerian ducts, okay? The external genitalia include the genital tubercle, the urethral fold, urethral groove, or the labial scrotal swellings. Depending on which type of hormones are present, either testosterone or anti-Mullerian hormone, those can differentiate into either the scrotum or the labial folds, right? And then the inside, they'll develop into the ovaries, the ovarian ducts, the vagina, or the vas deferens, the penis, right? So for males, if you develop a fetus, an embryo that is an embryo is fertilized, and it contains the Y chromosome, that chromosome is going to have this gene called the SRY gene. And that SRY gene is going to have the instructions to develop male sex characteristics. And so the production of testosterone is going to develop the Wolffian ducts, which become the epididymis, right? The vas deferens and the seminal vesicles, right? At the same time, those Mullerian ducts, which are structures for female characteristics, they're going to degenerate, right? And then the testes will develop later with the production of high levels of testosterone during puberty, okay? Same thing with females, right? If the embryo is fertilized with an XX, right, the lack of the SRY gene in that Y chromosome is going to allow the Mullerian ducts to develop into the upper portion of the vagina, the uterus, and the fallopian tubes, okay? I'm sorry, the Mullerian duct. And then the Wolffian ducts, which are typically ducts that are for male characteristics, are going to degenerate, right? So it's the opposite, right? And we know that the gametes, right, the gametes that these ovaries and testes are producing, they have different developmental stages, right? So for females, right, you are born with all the eggs you will have for the rest of your life, right? So they're kind of frozen in time until they're released and developed, okay? So you undergo meiosis when you're an embryo, and by the time you're born, those eggs are ready to go. They have already been shuffled around. Their DNA is shuffled around to give you different types of gene orientations, right? And they're just waiting for the reproductive age to start releasing those eggs. Whereas in males, right, they haven't gone through meiosis. they have only gone through mitosis. And so during puberty, that is when you start developing the variants of the genes, right? You start shuffling around the genes within each sperm cell to produce one type of genetic variation, right? So that way you don't produce twins and the same offspring all the time. Okay. Let's see. And then we talked about the hormones, the important hormones that are regulating the development of the reproduction organs, right? And those hormones include follicle stimulating hormone, luteinizing hormone, and gonadotropin releasing hormone. Which of these three hormones is released from the hypothalamus?
Gonadotropin releasing?
Yeah.
Gonadotropin releasing hormone, yeah. Exactly. And then I think we're going to stop there. Okay. Right here. Because that's already a lot. Okay. Any questions?
I think Alex might have a question. He put something in the chat.
Go ahead, Alex. Sorry, I didn't see the chat.
Oh,
Can you go over the chromosome a little, just like a little quick?
Yeah. What about the chromosomes is a little tricky?
You sort of lost me on the was it slide 7? 7.
This one? Okay.
Yeah.
So in this slide, we're talking about the sexual organs that are in the fetus. Okay. There's going to be these structures in the fetus that will develop either into male sex organs or female sex organs, right? And so let's say the fetus is a male, okay? And so the presence of the Y chromosome in all of the cells in that fetus are going to produce these chemicals and hormones like testosterone and anti-Mullerian hormone, which will then degrade those structures like the Mullerian ducts for that will become female structures, right? So Mullerian ducts are structures that all the fetuses have, right? But one of these two structures is going to survive depending on whether it's a male or female, okay? Mm-hmm. So if it's a male, it's going to destroy these Mullerian ducts and promote the growth of these Wolffian ducts, okay? And those Wolffian ducts become the epididymis, vas deferens, and seminal vesicles. And then later on, the testes will start dropping and forming, right? Now, if the fetus is a female, it's going to degrade these structures that are for males and promote the growth of these Mullerian ducts, right? And so depending on which of these ducts survives depends on whether you have an X or a Y chromosome, right? And so now let's talk about the case of the variants, right? Because we are dimorphic human beings that with dimorphic sexual characteristics, either male or female, but that doesn't mean that it always happens, okay? Sometimes you can have an XXY. So in this case, right, if you're an XXY, you are not only producing testosterone, but you are also producing AMH and estrogen that will support the production of the Mullerian ducts and the Wolffian ducts. So developmentally, you're going to have this kind of like in-between stage of development where you're developing both sexual characteristics. And that happens sometimes, right? That happens where a person is born with both male and female sexual organs, right? That are viable. Sometimes more than others, but there's a gradient. But for the most part, we are born either male or female, right? And this is just the diagram, the slide that's telling you these are the structures in a growing fetus that will become the sexual organs, right? And they depend on these genes, right? These male genes.
Thank you, Professor.
Yeah. All right. So I'll work on the exam today, and then we'll be ready for Wednesday. Okay.
Sounds
Do you know how
good.
many questions will be on there, Professor?
It'll probably just be like 40 questions, well, 10 per chapter.
He said probably.
Yeah, it just depends on what I could get in there, right? I don't want to, I don't want to give you too many. I don't want to give you too little, right? Because I gotta hit certain points.
Stick to what you talked about today, okay?
Yeah.
I
I'll try to.
know.
Yes.
Yeah.
Professor, please
just
be
Okay,
nice to us.
promise?
What were you saying?
Thank you. Oh, I said, Professor, please be nice to us. We got this, right? Oh my gosh.
I'm trying to be nice.
No, I know you are actually really nice.
I'll see you guys. Okay.
Thank
Have
you,
a good
Dr.
weekend.
Santana. We
Bye-bye.
appreciate you.
Thank
Bye-bye.
you. Take care. Bye, everyone.
Bye bye.
Bye.